right, great. Thanks, Jim. I like to think of myself as the algae czarina, I think it technically is. This is my first CABW, so it's a real pleasure to be here. Thanks, Jim, for the invite. And I'm excited to chat with you about the California Statewide Algae Plan. So as many of you are intimately familiar, California has been supporting the collection of algal bioassessment data throughout the state since 2008, and that has resulted in a large collection of algae taxonomy data that has been steadily growing over the past 8 years, as you can see here. And these campaigns have resulted in a rich database of information about the taxa that inhabit wadable streams throughout the state, and that includes, of course, both pristine states and those that have been heavily impacted by human activity. So the goals of the Statewide Algae Plan are to capitalize on this extensive knowledge base that we have about algal communities across the state and use them to develop tools that allow us to, of course, infer ecological condition and make informed decisions regarding the health of various ecosystems. So today I'll just be giving you a brief overview of some of our main goals for the Statewide Algae Plan, and this really circles back to what Pete was talking about yesterday, where we're focused on having standard methods and tools so that we can generate this data, have standardized ways of analyzing the data, ways that we can build efficiencies so that we can continually grow these multi-use data sets, and then figure out ways of interpreting these data using specific tools that then make it easy for regulators and policy decision makers to use the output of those important tools to make decisions about biointegrity and nutrient policies across the state. All right, so let's jump in with a little bit of algae propaganda. So why do we like using algae in bioassessment? So they are primary producers, and they are directly linked to nutrient concentrations and imbalances in our waterways. They are sensitive to different changes in chemistry. They have really short, rapid growth rates, which means that they respond really quickly to environmental perturbation. And of course, when we talk about algae species, we're talking about diatoms and soft-bodied algae and cyanobacteria, so that means we're including some of the potentially hazardous and toxic algae species, which means when we're looking at algae, we're not only inferring the biotic integrity of an environment, but we're also learning something about the potential human health implications. All right, so for many of you, these two documents hopefully look pretty familiar. Within the past 2 years, SWAMP has updated both the field algal bioassessment sampling protocols and the laboratory protocols for generating that important algae taxonomy data. Calvin gave us a nice introduction to where you can find those on the SWAMP website. And so for many of you, these sampling tools that we use for collecting algae out in the field might haunt you in your dreams, but they are very special to us because they allow us to generate biovolume estimates of different algae and their proportionate abundances present in the environment. So capitalizing on that big resource of algae taxonomy data that's been collected throughout California for the past number of years, in 2014, Betty Fetcher generated our first California Algal Index of Biotic Integrity, or the IBI, as we like to call it. And this index really focused on regions in Southern California. And it looked specifically at different groups of different algae taxa to see how they corresponded according to whether a site was considered stressed or considered in reference condition. So she looked at she compared whether you could generate an index using only diatom taxonomy data or only soft-bodied algae data, or actually a hybrid of the two, and she found that there was the most statistical power in separating those reference and impacted sites if you use this hybrid index that incorporates all those different algae taxa. So Andy Wren has done a nice job of comparing this algal index of biotic integrity with the index that we use based on benthic macroinvertebrates. So we call that one the California Stream Condition Index. It was developed by Rafi Mazur and others. You've heard about it a little bit already at this conference. And now that we have these two tools, Andy did an analysis to see when are they agreeing and when are they disagreeing in terms of biointegrity estimates for these different sites. Andy was also looking at the CRAM index, which is a rapid assessment protocol that looks at biotic structure and physical structure, but it doesn't specifically look at different biological species. And this graph here on the left shows you in green locations where those two indices, the one based on benthos and the one based on algae, when they agreed. And then in red, we see sites where they disagreed. And in general, what Andy found was that the CSCI was more sensitive to physical habitat conditions, whereas the IBI, or the algae-based index, was more sensitive to changes in chemistry. So what that really told us was that you have added power in combining those two indices and having multi-lines of evidence for interpreting the biological condition of a site. So two indices really are better than one. Moving forward. So one of the things about the statewide algae plan is that in a lot of ways we're trying to play catch up to the different bioassessment protocols and tools that have been developed using those benthic macroinvertebrates. So the California Stream Condition Index that was developed using those bugs is what's called a predictive index, and we were really jealous that they had this predictive index that could use all across the state of California. Because as I mentioned, the IBI that we have for algae is specifically tuned to Southern California. So moving forward, we are now developing a statewide algal index, which was referenced a little bit yesterday. And to do that, we're first generating a data set that capitalizes on all the different algae taxonomy data that has been collected through a number of different monitoring campaigns, which you can see listed here. It includes the SMC, the PSA, the RCMP, the RMC. Right now we're at about 2,000 stations, almost 4,000 different algae taxa are included in this data set. This number will probably grow. And I would like to say, if you have algae taxonomy data that somehow has not ended up in either the SWAMP or the SMC databases, please let me know. We would love to have it included, so come find me after. So one of the reasons we're really interested in developing this reference-based approach is that you can set these site-specific expectations for all of your different taxa that you find at different locations throughout the state based on whom you would expect to find there, given different reference locations. This metric, again, will be following the template that was used by the California Stream Condition Index, and that will have two separate parts. One of them is an observed over expected index, so that gives us an estimate of taxonomic completeness, something we heard about yesterday with Chuck Hawkins. And then the second part is more similar to the algal IBI that currently exists, where we develop this multi-metric index, and that gives us a gauge of different ecological structures. So what algae taxa are present, and what are some of their different ecological conditions that we tend to find them in? Are they phosphate-loving organisms? Are they sensitive to copper? Those sorts of ecological indicators. We are currently working on screening a number of sites to be included as our reference locations, and the screening criteria that we're using are listed over here. If they look familiar, it's again because it's the same screening criteria that they used for developing the California Stream Condition Index. So we're working hard at getting all of this together, and we expect it to be rolling out in fall of 2017, so be on the lookout for that. All right, so one of the other big goals of our statewide algae plan right now is addressing something that we call the capacity problem. So we love generating this great algal bioassessment data, but a lot of times we have samples that find their way to these very specialized algae taxonomy labs, and then there's a long wait time before you actually get some of that algae taxonomy data back. So how can we make it that we're generating all of this data as efficiently as possible? One of the approaches is developing what's called a standard taxonomic effort, or an STE. And with an STE, you basically have a standard taxa list and level of taxonomic resolution, and then taxonomists use that information and they can perform their analyses down to the level of taxonomic resolution that is specified by the STE. What this does is then minimize the time spent harmonizing our algae taxonomy data from different labs, and it speeds up the algae analyses. So in developing our STE, we're again modeling it after the SAFIT STE that was developed for freshwater invertebrates. And the way that we make decisions based on whether a different taxa will be analyzed down to the genus or the species level, we can use information based on if it's a really high or low abundant organism across the whole state of California, or if it has a specific trait that we want to make sure gets included that's specific to that taxa down to the species level, or if it's a really hard organism to ID, we might say drop that organism down to the genus level. So we have a working group that's going to be developing this STE over the next couple of months, and we should have a draft by June 2017. So that'll be exciting. One of the other ways that we're addressing our capacity problem is in the development of a molecular approach to algal bioassessment. So, like I said, a lot of time is being spent waiting around for algae taxonomy analyses because there is such a burden being put on these few and highly specialized algae taxonomists. So we are investigating a movement over to using DNA-based methods for gauging algal taxonomy from our different samples. So some of the highlights of this approach would be that it'll help us illuminate previously overlooked species if our DNA methods can be extra sensitive and find some organisms that previously we haven't been able to identify, potentially because of ambiguous morphology or just if they're novel species. There are dozens of commercial and academic labs that can perform these analyses, and for the most part, it's pretty inexpensive. The technology is just getting faster and cheaper, so it can be really appealing from that angle as well. In January or February, we rolled out a molecular sampling protocol, and many of you willingly volunteered to collect DNA samples for us, and we are eternally grateful. And we were able to send out a number of sampling kits to our collaborators, and the DNA protocol itself just involved collecting one extra filter from your composite bucket during algal bioassessment sampling, and then that filter was preserved in a preservative, which then keeps the sample stable, frozen for months to years. So we have a lot of flexibility in terms of if you're out in the field and you can't get your sample to the freezer that same day, you have a little bit more leeway. And we are really excited about getting those DNA samples back from our collaborators. So if you have samples sitting in your freezer, come find me after, and we're really excited to get our hands on those. So the key questions that we have for our initial pilot studies looking at the DNA sequence data of these algae samples is how do the morphology-based and DNA-based algae taxonomy data compare? Are we picking up the same organisms in similar relative abundances? What algae species that we're finding in the DNA data are actually missing from DNA reference databases? Do we have some really important taxa in the state of California DNA sequenced yet? Do we need to go back into the field, grab a sample, and sequence it ourselves? And how well are our algal indices going to perform with this DNA data? That's the big question. That's something we really want to verify. And we have this in mind as we're developing this new statewide algal index, that at some point we might want to be using it with DNA-derived data. So that's a big priority for us. We expect to have the results of these pilot projects by about early 2018. All right, so in the last few minutes, I just want to talk a little bit about what we envision for the future. So now that we have these standardized protocols in place, we're generating this really awesome algae taxonomy data. We want to make sure that the tools that we have in place are able to translate that data into decision-making tools as well. So some of the ways that we envision being able to do this is incorporating the forthcoming statewide algal index into online resources. So both through the SWAMP portal, but also have a standalone resource where if you're generating taxonomy data, you'll then be able to calculate your algal indices. We also envision building a California algae DNA reference database. So what does that mean? So that would be a collection of reference DNA sequences that correspond to specifically the algae that we find in the state of California. These figures here might look familiar. They're snapshots of the taxonomy resource websites that were created by CSU San Marcos and others. They're a really great tool if you haven't checked them out before in terms of identifying California-specific algae species. So what we envision is being able to build on top of these existing websites and link them to the DNA for each of these different California-specific algae as well. And lastly, of course, we'll be focusing on developing some guidance documents to help you analyze both your algal taxonomy data, generate those important algal indices, and then also interpret your DNA data. And lastly, just a couple questions that we like having in the forefront of our minds when we're thinking about how to implement these tools into regulatory decisions and to share these informations with our collaborators and different state and local partners. So how will our algal toolkit support State Water Board's combined nutrient and biointegrity policy? So how are we going to be using this algal bioassessment data to set nutrient thresholds? How are we going to use it with other multiple lines of evidence, such as the California Stream Condition Index, and how are we going to interpret those indices together? And lastly, we have a little bit of a placeholder bullet point where it just says, are there any required technical elements for implementation? So what elements are we forgetting about? Are we missing? Are we going to have to be developing in the future so that all of our algae taxonomy data is finding its ways to the user community as efficiently as possible and really giving everyone the tools that they need to interpret that taxonomy data into useful decision making? So of course, all of the work that you saw presented here has really been built upon the foundation of great bioassessment work that's been done at the state. And feel free to contact me if you have any questions about anything I spoke about today, but especially if you have sampling locations where you would like to pair algae morphology samples with algae DNA samples, let me know. We'd be happy to send you a sampling kit and have you on board. And also if you, as I mentioned, have algae bioassessment data that hasn't found its way into the SWAMP database, please find me after, and we'd love to get our hands on that. So thanks a lot for your time.
Okay, one quick question.
Yeah, so you mentioned the standard taxonomic effort that's going on. However, I'm not aware of any working group like SAFIT or SCAMIT that's currently going on with the algae. It's something I've been asking about for years now. So what's going on with that? How do we join? How do we become active members and actually clean up the seeding list with all of the multiple provisionals that, and you know everything that's going on with it as well as I do. So how do we become involved and join the group?
It's great to hear that people are so excited. So we're in the preliminary planning stages. So we've partnered, so SWRCB and Cal State San Marcos, we've had a couple meetings so far where we're drawing up our lists of interested parties. So first there will be the experts that we contact about brainstorming ways to actually develop the nuts and bolts of that first draft of the STE. But I'll keep you in mind when we begin rolling some of those drafts out.
Should it be more inclusive of the STD? More robust list inside a box?
So in terms of incorporating with other agencies, our experts will come from a wide variety of different agencies. So that would include Rithron and EcoAnalyst, and for sure some of our frequent collaborators. But I hear you, and that's a great thing to keep in mind. Thanks.
Thank you again.
Thanks.